But first, I wanted to very quickly give some information on how this seminar series came about. And with this, let me first present our the MetaBrain project that we are also part of. This is a European Innovation Council funded program for 3 years. It's 8 partners from 5 countries, and the coordinator is INDIBAPS in Spain. And we, as the Cyprus Institute of Neurology and Genetics, we joined through the Hobon facility last year. And the focus is magnetoelectric and ultrasonic technologies for advanced brain modulation. And this is a very interdisciplinary consortium that combines experiments and computational modeling in order to try and achieve this very precise spatio-temporal control of brain activity. You can also see the team that works, the local team of MetaBrain here. Rafael, Agisilaos, Dimitris, Stella, and Katerina. We are part of the institute, the CING, as we call it in brief, which is a center of excellence for clinical services, research, and education. And we are part of the bioinformatics department, which focuses in systems bioinformatics within the precision and personalized medicine area. Now, let me switch to our speaker, Professor Marcus Kaiser, who will be giving the first lecture. Let me just stop sharing. And Marcus is with us today virtually from the University of Nottingham. He's a professor there and a co-director of a new and exciting center that he will be talking to us about on neurotechnology, neuromodulation, and neurotherapeutics. So, Marcus, thank you for joining us, and the floor is yours.
Yeah. Okay, thanks for the invitation and for the introduction. I'm based in the School of Medicine, but my background is from computational work and then moving more into experimental work with medical applications. If you know one thing about Nottingham, you know about Robin Hood. If you know 2 things about Nottingham, you know that MRI was invented at Nottingham University, leading to the Nobel Prize to Sir Peter Mansfield. And because of this history of medical imaging at Nottingham University, we're also the National Center for High Field MRI. So we're getting an 11.7 Tesla MRI scanner, which means that we can look at the human brain in much higher spatial resolution. So the system is currently being installed, and we're very interested to see what kind of information we can get in healthy participants and in patients. We also just started a Center for New Technology, New Modulation, and New Therapeutics. It's cross-faculty with around 30 faculty members. And we can go all the way from mathematical and computational modeling to developing novel devices to animal studies and to clinical research. So what you see on the left side of that image there is a mobile MEG system called OPM MEG. And because it's mobile, you can freely move the head during measurements. So it's ideal for measurements in patients, ideal for measurements in children. There's a startup company, Circa Magnetics, that is developing this device. And on the right-hand side, you see ultrasound, which is the focus of the talk today. So what is the demand for novel technologies for brain stimulation? The demand for non-invasive, personalized, and targeted neuromodulation. We had a survey with around 800 participants, half of them healthy and half of them suffering from brain or mental health conditions. Many of the patients had Parkinson's disease, but there were other conditions as well, like depression and anxiety and so on. And we were asking them, what kind of intervention would you prefer? We gave them 5 different options, from pharmaceutical drugs to brain implants to different kinds of non-invasive brain stimulation. And their first choice was ultrasound. In terms of preference, people possibly thought about pregnancy scans. So if it's safe for pregnancy scans, it's safe for brain interventions. Then we have magnetic brain stimulation, pharmaceutical drugs, electric brain stimulation. people possibly thought about electroshocks, so that's not very popular. But on the last place, you have brain implants, with many people saying that they would never use brain implants. So both for patients and for healthy participants, there were many saying that they would never use them. Why was there a preference for ultrasound? It's based on the perceived safety of the device. So the safety of ultrasound was seen as much better than for other interventions. In terms of the effectiveness of an intervention, ultrasound and implants were seen as having the same effectiveness. So implants, of course, you can stick an electrode into one part of the brain. So it can be very effective because you're directly at the target. But because of the safety, the preference was for ultrasound. So how does ultrasound work? You basically have a sound wave, a mechanical wave that is going through the skull, and the sound source, the transducer, is a bit curved, which means that the wave is focusing on one particular spot inside the brain, the focal spot of ultrasound. So at this focal spot, you have a higher pressure change because of the ultrasound, and this change is leading to mechanical deformation of actins inside cells. It's leading to activation of mechanosensitive channels and can then lead to second messenger changes, calcium changes, and internal processes that then change the probability that channels are opening. So this is a device that came onto the market for studies in humans, came on the market in October 2019. So we can directly stimulate deep brain structures up to 8 centimeters deep. There are several centers in the UK that have those kinds of devices. We have 3 devices here in Nottingham, and there are other centers, and there are more and more centers basically purchasing those devices. The focal area for the device on the left is 1 to 2 centimeters. There are other devices that cost 10 times as much, and they could have a resolution of 1 to 2 millimeters. But most labs have the device that you can see here with a 1 centimeter resolution. Since focal, we can go directly for deep brain structures like amygdala, thalamus, hippocampus, basal ganglia, cerebellum. It's non-invasive. Depending on the protocol, we can either increase or decrease brain activity. And the device could be used in humans, or for other devices can be used in rodents or rhesus monkeys. So it's really flexible what you can do with those devices. And interestingly, you can measure changes in brain activity at the same time. So you can measure changes in MRI, the device is MRI compatible, or you can measure changes in EEG while you're stimulating. So there are different ways how to use the device. You could use it offline. Offline means that you stimulate and then you measure effects after ultrasound stimulation. So you stimulate first and then you roll people into an MRI scanner, for example. Alternatively, you could have online stimulation, so you measure effects during stimulation. This could be having an EEG cap on while you stimulate with ultrasound, or it could be at the bottom using ultrasound stimulation while participants are inside an MRI scanner, and then you can directly observe fMRI changes while you are stimulating. So that's a video from Nottingham here where we stimulate with ultrasound while the person is in the scanner. And just to give you an idea what might be possible in the future, at the moment, if you have people with Parkinson's disease, with essential tremor, you basically have the option to have an implant or you remove part of the brain tissue. But this study here shows an example where you can use ultrasound brain stimulation to get rid of the tremor. So this is a device that is the more expensive one, so 10 times more expensive than our device, where you are inside an MRI scanner and you can stimulate a very small part of the brain. And in this study here, for at least 25 minutes, you have a 98% reduction in tremor in those patients. Okay. Why only 25%? Because the surgeons wanted to start destroying brain tissue. So they waited for 25 minutes and then they went ahead with their usual procedure, which is turning up the power a lot more and destroying brain tissue. But it's a proof of principle that ultrasound can get rid of the tremor non-invasively. So why not just use invasive technology? Why not use a different kind of technology? So the reason is that for each operation, for each surgery, you have an infection risk of 4.5%. And just need to, yeah, okay. So this is infection risk of 4.5%. And if you have an implant, you might need to change the implant over time. So if you have an implant for 40 years, you might need to get back and have another surgery to replace batteries or replace electrodes and so on. So if you have multiple surgeries for replacement for 5 surgeries, there's a 20% chance that at least one surgery goes bad and you do have an infection. So this is not going to happen. Well, let me just quickly check. Yeah. Okay. So there are other reasons why invasive implants are not a good idea for the future. One is that there are always those potential side effects. So you might get approval to have a brain implant in adult patients, but it's very unlikely to get any ethics approval in children. And it's extremely unlikely to get an approval to have implants in healthy participants. And the total addressable market for invasive new technology is limited. So maybe only 6% of patients can get an implant. And the market size will decrease further if other opportunities become available, like digital, cognitive, or non-invasive tools. The currently addressed market share is much smaller. So only 0.01% of patients actually benefit from brain implants. So even if you look at applications such as deep brain stimulation devices, DBS devices, and Parkinson's disease, it's only 1.4% of patients that have those devices. The market share or the number of patients that are benefiting is increasing slowly, but then you also have the limit that there were only few centers that can perform brain surgery. So in the UK, there are only 12 hospitals that can implant brain devices. So there are only 400 procedures every year in the UK for having brain implants. So there's a real limit on this kind of technology. So rather than brain implants being a gold mine, in my opinion, they are more a graveyard for new technology. So let's look at target effects of focused ultrasound neuromodulation. So this gives you an overview of studies in human participants. So both in healthy human participants and in clinical studies. And you can see that a range of conditions has been tested and a range of targets has been tested. So this gives you an overview of clinical studies. So you can see ultrasound studies in Alzheimer's disease, in generalized anxiety disorder, schizophrenia, Parkinson's disease, and many other conditions. And we have this overview of ultrasound studies in humans, so healthy participants and clinical studies. And we also have an online database to add your own study. We were looking at the effect of different parameters. So in theory, if you have a higher intensity, a lower duration, a higher duty cycle, the idea is that you would have an enhancement of brain activity. On the other hand, for lower intensity, the idea is that you have reduced activity in your target structure. So we did a systematic literature review of different studies that are out there. And this shows you the result for those studies. So we have in blue a reported enhancement of brain activity and in red a reported reduction in brain activity. And the blue line tells you what would be expected from a theory in the field. So the expectation from the neuronal intramembrane cavitation excitation models. And you can see that the results don't follow the model. Okay, so in theory, everything on the left side under the curve should be suppression, but you can see that there's enhancement as well. And for online studies, you also see that there was suppression on the right-hand side of the curve. So there are additional properties that are not part of those biophysical models. The idea is that the response not only depends on membrane biophysics, but it also depends on changes in individual brain regions. So there might be a different balance between excitatory and inhibitory neurons, and because of that, there might be some changes in the effects of ultrasound brain stimulation. I'm just quickly checking. Margarita, are you still there, or are there any Wi-Fi issues?
We can hear you perfectly.
Yes. Okay.
It's
Excellent.
uninterrupted.
Yes.
Yeah.
Because I did have some Wi-Fi issues yesterday, so I'm just checking. Okay. So let's look at network effects of focused ultrasound. So we're interested in what is happening not just at the target region, but also throughout the network. So what you see here is the brain network based on MRI recordings. You have a special protocol in the MRI scanner called diffusion tensor imaging, which allows you to reconstruct connectivity in the human brain. So the color here shows you the local direction of a fiber tract. Green means from the back of the brain to the front of the brain, or the other direction. Red means left to right or right to left. And blue means top to bottom or bottom to top. So unfortunately, we don't know what the direction of the fiber is. We just assume it's bidirectional, but that might not be the case. And actually, animal studies show that we have some one-way streets in the system. But what the data shows us is just some bidirectional. We just assume it's bidirectional, even though we know it's not always bidirectional. So we can look at this network, and we can already predict age and intelligence based on brain connectivity. So age plus minus 8 years, and intelligence plus minus 5 IQ points. But the idea is to use it as a starting point to predict what actually happens if we stimulate one node, one region in the network. So just to give you an idea of computational models that we can use. So this is a model developed for epileptic tissue for epilepsy patients, where we have the connectivity between different parts of the brain, like in the previous picture. And for each brain region, we have a dynamical model that is simulating oscillations in the brain region. And in this case, we have small oscillations, but then over time, the amplitude of oscillations is increasing. And once we have large-scale amplitudes in a brain region, we would say that we have seizure-like activity in that region. So we call that the escape time, the time point when we see those large-scale oscillations in the system. So we can basically model which parts of the brain are escaping earlier than others. And we do find that the areas that escape early are also the areas that are known to be starting points for epileptic seizures. We can use this kind of model to look at patients that are undergoing surgery, epilepsy patients. So they have temporal lobe epilepsy, and they have implanted electrodes. So you can see at the top that you have a grid of electrodes that is implanted on top of the brain under the skull. So we can basically record the activity that is going on. We can look at functional connectivity between electrodes. We can reconstruct the functional network in the system, and we can then run dynamical models on the functional network. So this is what you see in the center of this figure, where we have simulated dynamics and some parts of the network escaping much earlier. So those could be potential starting points of epileptic seizures, whereas other parts are escaping much later. But the interesting point of this is that the functional network is based on the non-seizure activity in the system. So we don't need any seizure to occur in our computer model. We just have the implant, and we just measure what's going on when the patient is in a healthy state, when there's no seizure going on. So that's our starting point. We don't need to wait hours or days for seizures to occur. We can get the background activity that is going on as a starting point for our model. So the non-seizure activity in the system. And we can then use this to predict whether surgery is working or not. So we can have a comparison between what the surgeons were removing, so that's the tissue under the black electrodes, and in color you see what our computer model is predicting in terms of the starting point of epileptic seizures. So on the left-hand side, there is a complete overlap between what the surgeons are removing and what we are predicting as being the starting point of seizures. On the right-hand side, there's no overlap at all between what the surgeons removed and what we see as a potential starting point. And if you then go back to the patient, the one on the left with complete overlap was seizure-free after surgery, and the one on the right was not seizure-free. So how often does this happen? If our computer model says that the patient is seizure-free, then in 100% of the cases, the patient is seizure-free. If our computer model says that the patient is not seizure-free, then in 73% of the cases, the patient is not seizure-free, and in 27% of the cases, the patient is seizure-free. So we get a very good prediction in terms of surgery going wrong, in terms of having surgery and the patient still having epileptic seizures. So this computer model could be used in the future to predict whether surgery works or not. And it also could be used to predict alternative targets for brain surgery. And at the moment, the two people who were involved in the study, Peter Taylor and Yujia Wang, they both have UKI Future Leader Fellowships. So they continue to work on those computational models and predicting surgery effects in patients. And we can also use Kuramoto oscillators to simulate activity in the system. So this is a study in healthy participants. Each brain region is represented as a Kuramoto oscillator. So we basically have a certain frequency in the network. Let's say all brain regions have a 40 Hertz oscillation. But then the phase of the oscillation can be different between the regions. We also have coupling between the regions. So the stronger the fiber tract, the stronger the coupling between the regions. And we also have delays between the regions. So the longer the fiber tract, the longer is the delay in the system. Mathematicians really don't like delays because it makes things more difficult. But we thought we need delays, they are important. So we have delays in our computer model. We can then measure the synchrony over time in our computer model. So we have the mean synchrony, and we also have the variability, the fluctuation of the synchrony over time, which is the metastability. And what we basically showed in this paper is that the brain has a higher metastability than would be expected. So higher metastability makes it easier to switch to different brain states. So if you have higher fluctuation, it's easier to go to different states than if you have very narrow fluctuation. So you have more fluctuation in the brain than expected. And this also links to brain diseases. We know from dementia patients that they tend to get stuck in a brain state. So they have lower fluctuation. They are stuck in a state for longer. It takes them a longer time to switch to a different brain state. So metastability is very important. So then we wondered, can we actually use those dynamical models to predict intelligence? Okay, so can we link that to intelligence? So in this study here, which we're currently preparing under review, we looked at topological measures and the link between network topology and intelligence. So you see the full IQ, crystallized intelligence, and then different kinds of fluid intelligence, processing speed, visual spatial ability, and memory. So you can see correlations here, you can see significance levels here. And then we have different network measures. We have clustering coefficient for unweighted and weighted networks, characteristic path length for unweighted and weighted networks, and generic edge density in the system. So you can see that some features are correlating with cognition, but very few and not strongly. We can then look at spatial features, so the convex hull of the whole brain. We can look at distances in the network. So if you have a triplet A, B, C, and A is connected to B and C, we can basically check, is A connecting regions B and C that are nearby, or are B and C far away? And also, are the connections A, B, and AC, are those short distance or long distance connections? And then we can also look at the metric path length. So we're calculating shortest paths, but we're calculating the distance over the shortest paths in terms of the length going through those fiber tracts. And you can see that there are many cognitive features that are strongly correlated with spatial features. And the correlation is stronger as well. We can then also look at the dynamical models, the Kuramoto oscillators. We can have different oscillations going on, 40 hertz, so gamma range, 20 hertz beta, and 10 hertz for alpha. And we see a stronger correlation for gamma, for 40 hertz frequency. But in general, we see many strong correlations between dynamical network features and intelligence. So it could be something for the future to look at those dynamical features rather than only look at network topology. So looking at dynamics and looking at spatial features to link this to cognition. So let's get back to ultrasound stimulation. So we're trying to link those network features with effects of ultrasound stimulation. So what you see here is a study in healthy human participants where we have resting state, fMRI, resting state brain activity as a baseline, and then we have a stop signal task. So you see a green circle, and once the green circle appears, you're supposed to push a button. But if you have a red cross appearing, you should not press the button. Okay, so you have to inhibit the button pressing process. It's really, really difficult. It's really annoying. So I was one of the participants. So it's a really annoying task. You have a high error rate. Okay, so very often you see the circle and then you push the button even though the red cross is appearing. We then plan our ultrasound experiment, and then we have a second session where we have ultrasound stimulation first. Then we have MRI scan again. We look at changes in brain activity, and then afterwards we have that stop signal task. So the stop signal task happens 1.5 hours after ultrasound stimulation. And we have two different brain regions that we're targeting. It's either the thalamus, the white thalamus, or the white inferior frontal cortex that we're stimulating with ultrasound. And we basically stimulate with those parameters. But the most important bit is that we have a duty cycle of 10%. So we only stimulate 10% of the time. And in total, we only stimulate for 80 seconds. Okay. So 10% means that effectively the active stimulation is only 8 seconds. Okay. So we have 8 seconds of ultrasound stimulation, and then we check what changes we observe. So even though it's only 8 seconds, we do see changes in functional connectivity for both targets, for inferior frontal cortex and thalamus. So you can see those changes 20 to 50 minutes after ultrasound stimulation when people are inside the MRI scanner. So on the left-hand side, you see some connections that show reduced connectivity strength in blue. On the right-hand side, you see for the thalamus some connections that are reduced in blue, but other connections are increased in red color, including the contralateral thalamus on the left side. And you can also see that those changes occur at different time steps. So some parts of the brain show changes that are earlier than other parts of the brain. So then the next question is, do those biological changes also correspond to cognitive changes? So first of all, we looked at those cognitive changes. If we look at the accuracy, we find a similar accuracy before and after ultrasound stimulation. But for ultrasound stimulation, we find a faster reaction time for inferior frontal cortex stimulation on the left side. So if you look at the pre-stimulation and the post-stimulation, we see 8% faster reaction time after ultrasound stimulation. For stimulating the thalamus, we don't see a change in reaction time. So it's only the inferior frontal cortex where people get 8% faster. So you might say 8% faster is not much, but for some applications, let's say you're in a fighter jet and you're 8% faster pushing a button, it might make a difference between getting shot down or not in a fighter jet. Okay, so there are some applications where 8% faster is really important. And also 8% faster is 1.5 hours after stimulation. So maybe if we would measure 20 minutes after stimulation rather than 1.5 hours after stimulation, maybe reaction time is even faster. So that's something to test in the future. And we can link this to biological changes. So if we look at inferior frontal cortex, the reaction time change is directly correlated to the change in functional connectivity that we see. And is also directly related to the amount of pressure that we can get to the target. And that's some mediation analysis. So this basically means that we can predict the cognitive change in reaction time based on the pressure change at the target and based on the functional connectivity change that we see in the MRI. We're also using some information theoretical measures to predict effects in the system. We use redundancy and synergy. So only very briefly show this, but you can see more results in those papers here. Redundancy tells you what is information content in terms of repeated information that we can obtain on any variables, where each of these variables represent a measure of brain activity for a number of brain regions. So for example, fMRI bold signals. So the idea is that redundancy is very high if a lot of different brain regions do the same thing. Okay, so if the time series is very similar, you would have very high redundancy in the system. On the other hand, you can get information content that is exclusive to observing different brain regions. Okay, so if you have high synergy, it means that you get a lot of extra information by observing multiple brain regions. So we're looking at this in terms of changes in brain activity. So actually the one looking at synergy and redundancy is the one on the top right side, the network neuroscience paper, where we observe characteristic changes. But what we see here is just the basic change in functional connectivity for 3 different targets in rhesus monkeys. If we just look at the first target here, anterior medial prefrontal cortex, we basically can observe those different curves here. And then it depends on what time point we are looking at. So if we observe changes early on, we can basically see a peak here for the first brain region. Okay, sorry, just to go back 1 second. Okay, so we stimulate AMPFC, and then we observe changes in 3 other brain regions that are connected to that one. Okay, so this shows you the functional connectivity changes in those other 3 brain regions. And for the first brain regions, we see an early change. For the second one, we see a change a bit later on. And for the third one, we see a peak later on in the time series. Okay. But the challenge is that if you look at 3 different labs, maybe one lab is observing changes early on, and another lab is observing changes at the very end. So the first lab would say that, okay, the first brain region shows a change, and the third one doesn't show a change. Whereas the other lab would say, well, the first brain region doesn't show a change, and the third brain region is showing a change. So what you observe depends on the time point when you are observing. So because of that, it's really important to observe what is going on over a long time period. It's not enough just to take a single snapshot. You have to observe over a longer time frame to really catch the changes that are going on in the system. Okay. So I'm going to just briefly skip this one. And so what I was showing before was changes with transcranial ultrasound stimulation. So those are very expensive devices. The whole set is $200,000. And those are not devices that you can take home. And of course, you also need an MRI scanner for some basic measurements. So this approach doesn't really scale at the moment. So because of that, I'm also involved with devices that you can take home. So this is a device that is stimulating the vagus nerve around the ear. And as a disclaimer, I'm on the scientific advisory board of this company that developed the device. So we basically have the vagus nerve that is innervating the ear here, around the ear. And we have this ultrasound stimulation device. So on the right-hand side, you see the device. You can basically buy it for around $400 online. It's on the market as a wellness device. It's stimulating up to 30 minutes as a safety limit. We don't want people to fall asleep and then it's on for 8 hours. You basically have the duty cycle, intensity, and so on. So we can basically directly stimulate the vagus nerve. So this is a pilot study that was published last year where we have 30 anxiety patients. They have a higher anxiety score as measured by the Beck Anxiety Inventory. And they just take the device home. They use it 5 minutes per day so they can decide by themselves when they want to use it. And 85% show a significant reduction in their anxiety score. So this is what happens. The blue measures show you the baseline before stimulation, and then after each week of stimulation, they do the assessment again. So after the first week, you already see a significant reduction, 30% for the anxiety score, and after 2 weeks, it's a 50% reduction. But you also see reductions in other scores like post-traumatic stress disorder, depression, and sleep. And this is the curve that is including participants where it doesn't work at all. Okay. So you see this increase even though 15% are not responding. And for the 15% that are not responding, we are not totally sure why. It might be they have a strange ear shape. So maybe there's no good contact. Maybe they didn't put in the ultrasound gel correctly, because if you put the device on like a headphone, then the gel is in the hair, not in the ear. So there might be some reasons why it's not working for the 15%. But 85% is responding is already a lot better compared to electrical, where only half are responding. That's a really promising approach. And we also now have a sham device that people can't distinguish the active device and the sham device. So we can now do randomly controlled trials. So we're currently running a follow-on study in anxiety patients where we have a control group as well. And we're also running studies in tinnitus patients. And there was one study starting in depression patients and one starting in chronic pain. So we're basically testing different effects of ultrasound vagus nerve stimulation. So this is just show you what might be possible for depression patients. So this is a study in healthy participants, but we want to move on to patients in the next couple of weeks. And for the healthy participants, you see that the emotional bias score is improving after ultrasound stimulation. So basically, the higher the score, the happier people are. So you have the baseline, you have in green the change for ultrasound stimulation in dark green, and then you have the sham in light green. So you see this strong effect already during stimulation and also after stimulation for ultrasound vagus nerve stimulation. And the effect is weaker and slower for electrical vagus nerve stimulation. So it's a promising approach to look into. And the future for brain stimulation is closed loop. So rather than just turning on the device, the idea is that you can take into account what the current state of the system is and then adapt the stimulation. So you can either change the stimulation based on what the phase of the oscillation is in the brain. So you basically make sure you're in phase, or you can measure whether stimulation is working or not. So if not, it's not working, maybe you turn stimulation up a bit or you stimulate a bit longer. If it's already working, maybe you just stop a bit earlier with stimulating. So those are some pilot studies here at Nottingham, where you see that the effect of a brain stimulation depends on the ongoing phase. So you can basically entrain the brain with transcranial alternative current stimulation, and then you can stimulate with TMS either in phase or out of phase. and the effect depends on your timing. So we currently have an MRC program going for the next 5 years, where we're basically developing this technology to have closed loop transcranial magnetic stimulation, but also closed loop ultrasound stimulation. We initially try this in healthy participants, so that's what we're doing for the next 1.5 years. And for the following years, we're basically looking at patients for depression and schizophrenia. We look at cognition, like working memory. And for Tourette syndrome, we look at motor function. So we're basically testing whether this closed loop stimulation is leading to better results in patients compared to open loop stimulation. So in summary, ultrasound neuromodulation is preferred by patients and by the public. Transcranial ultrasound neuromodulation effects are predominantly time constrained. They're spatially distributed to other brain regions, and especially to other brain regions that are functionally linked to the stimulated region. Biological effects are indicative of cognitive effects like faster reaction time, and those effects are still there 1 hour after stimulating for 8 seconds. For ultrasound vagus nerve stimulation, it's painless. So compared to electrical, you don't have any pain or itchiness on the skin. And it shows a high responder rate for anxiety, also for emotional bias tasks and other memory tasks in healthy participants. So there are preprints online. And we don't need an MRI scan, which is really nice. So ultrasound vagus nerve stimulation improves mood, as shown earlier, but it also improves semantic working in associative memories. So there are some preprints online on bioRxiv and medRxiv. So what we want to develop in the future is an ultrasound device that you can take home and that goes for the skull as well. So I started a company a few months ago, Deep Brain, where we are developing a device that you can take home. You don't need an MRI scanner, and you have this device at home, and you can go through the skull and directly stimulate deep brain structures. So it's early stages at the moment. We're just searching for some seed capital and filing some patents and so on. But we have some ideas how to get around the bottlenecks. There's also the book here, Changing Connectomes by MIT Press, where I discuss how we can analyze brain connectivity, how we can look at network dynamics, and how we can assess the effects of brain stimulation in the human brain and in animal studies. So there's a lot more information in the book here. So finally, I would like to thank my team, a current team in the lab on the left side, and some of the alumni in my lab who moved on to faculty positions. I would like to thank collaborators, the funders, and you can follow me on Blue Sky or X, Connectome Lab, if you're interested in more information. So thanks for your attention, and yeah, let's go for questions.